H3K4 demethylase. And when it was first discovered, it was part of a multi-subunit chromatin modifying complex. And one complex member that KDM1A associates with particularly strongly is called RCOR1. So when this was discovered, we immediately knew to look for a neurological function for this protein based on its association with RCOR1, and that RCOR1 linking KDM1A to an even larger complex that is well known to be important in neurodevelopmental disorders and in neurodevelopment in general. So that's looking interesting. And then a final piece of the puzzle why we think this enzyme is so important is that we know about some drugs that can target it. So we have a chemical angle. So we can come at this with a chemical biology approach. So I told you that RCOR1 is known to be important for neurodevelopment. And here are some of the types of experiments that people do to understand this and now extend this not only to RCOR1 but also to KDM1A. So RCOR1 complexes are known to bind to neuronal specific genes. These are things like ion channels and neurotransmitter metabolism genes. And in progenitor cells, it keeps them silenced. So these are cells that are almost neurons but haven't quite committed yet. And then when the cell commits to becoming a neuron, the brakes need to come off in a coordinated manner in order for development to occur properly. So here I choose some more visually striking examples of how this looks. So here we see the cortex of a developing mouse, and the new neurons are born at the basal layer, and they proliferate and then migrate up to the correct position in the cortex. And if you get rid of KDM1A using RNAi methods, you see that neurons fail to migrate to the proper position. In addition to that, there's a few different variants of KDM1A that are found in neurons. In an inactive null version, you see that neurons fail to arborize. They fail to send out processes that are useful for making connections with other neurons. Whereas in a constitutively active form, there's actually too many processes now that are formed. This is kind of an extreme end of this phenotype. So these are very critical early developmental biological processes. And so it's perhaps then not surprising that if you genetically get rid of KDM1A, that is as lethal at the embryonic stage and might fail to develop beyond day 9. So that's if you completely get rid of KDM1A, which is a bit extreme. In humans, we know of 3 patients that have mutations in KDM1A, not completely getting rid of it, but just point mutations, so single amino acid changes. And those patients have quite a lot less myelin, the lipid that coats neurons and increases connectivity, and they have significant cognitive impairment along with some other neuroskeletal and developmental issues. So absolutely KDM1A is critical for neuron development, but it's also expressed throughout the brain in adulthood, and it continues to have presumably similar functions of mediating cell proliferation, probably less migration, because that's less of a behavior that has to happen, although not during neurogenesis. And so this is an interesting target from a neurobiological perspective for a neurodevelopmental. So how does KDM1A do all this? Well, if we look at its structure, we can get a few clues. So I told you it interacts with RCOR1. It does this via its tower domain, which is this large insertion that mediates binding to a large number of co-regulators, RCOR1 being one of the most tightly associated, most prevalent binding partners. In addition to this tower domain, KDM1A has a swarm domain, which is highly evolutionary conserved among proteins that bind chromatin. And this is also likely to mediate protein-protein interactions with these large multi-subunit protein complexes. But then where the chemical biology gets interesting is the amine oxidase domain. This is the catalytic core of this enzyme, and it's what gives this enzyme its H3K4 demethylase activity. And it's highly homologous with the catalytic center of the monoamine oxidases, which I'll tell you a little bit more about in a second. And in colored sphere representation here, you can see the cofactor that KDM1A uses to do its catalysis. This is a flavin adenine dinucleotide, or FAD cofactor. So the monoamine oxidases have a very similar catalytic structure, and neurobiologists know all about them because they are the primary metabolizers of neurotransmitters and have been the targets of antidepressants and other neuroactive drugs for decades. In fact, the very first antidepressants we discovered targets the monoamine oxidases. There's two isoforms, A and B. MAO-A is primarily associated with the mood-regulating aspects of MAO-A pharmacology and is targeted for depression and anxiety, whereas MAO-B, highly similar enzyme, metabolizes a slightly different set of neurotransmitters, and it's associated more with cognition and is therapeutically targeted in Parkinson's disease. They both use an FAD cofactor to do their chemistry, which is shown here in a clearer schematic than with the protein structure. And there are a large number of drugs, including the antidepressant tranylcypromine, which I'll abbreviate TCP, that are relatively non-selective between the monoamine oxidases. And drugs like tranylcypromine target the FAD cofactor, and they form these covalent adducts to irreversibly inactivate the enzyme. And a part of this irreversible inactivation comes some metabolic issues that come with inhibiting monoamine oxidases with drugs like tranylcypromine. So although it's a very clinically effective drug, it seems to actually have pretty good efficacy as an antidepressant. It's not very safe to take. You have to be on a very restricted diet. And so as a result, this is considered an old drug that we really only bring out to treat very otherwise treatment-resistant depression because of the safety issue. So now if we look at any decorations that are on the amine oxidase domains of these proteins, you just see two little helices, and these are responsible for allowing the monoamine oxidases to attach to the mitochondrial membrane, which is where these are found. And then in contrast, there are the nuclear amine oxidases. I've already told you a fair bit about KDM1A. It also has a homolog that we've more recently discovered, KDM1B. Much less is known about KDM1B, and its role in the brain is unknown. So I'm not going to tell you very much about it, but it's there. And both KDM1A and 1B take the same catalytic machine that the monoamine oxidases have, but they use their extra domains, for example, the tower and swarm domains, to bring that catalytic machinery into nuclear complexes and to regulate gene expression. But all four amine oxidases use FAD, and they're all inhibited by tranylcypromine. So this is a good starting point. So here's another view of KDM1A with its FAD cofactor, and here is actually a structure of TCP forming a covalent adduct with FAD. And this inactivates the enzyme. So my preferred way of measuring this is using a mass spectrometry assay. What you can do is give the N-terminal tail peptide of the histone substrate, which is very disordered in the nucleosome, and treat it with recombinant protein. And KDM1A will use its amine oxidase chemistry to demethylate from the dimethyl to the monomethyl group, which is a very small difference in substrate, but no problem for a mass spectrometer to detect. And then you can add in a small molecule such as TCP and measure the inhibition of that demethylation reaction. And when you do that in vitro, what you find is that it's true, TCP does inhibit KDM1A, but it's not a very strong inhibitor. It's time dependent, but at a fixed time point, we can compare all inhibitors against each other. You just get in a standardized way. You get an apparent IC50 value in this case of about 50 micromolar. So this is several orders of magnitude greater than what you would like to see for a drug, but it's a starting point. But we asked whether this was really the IC50 in the brain. I told you that KDM1A is not a free enzyme like it is in a test tube. It's part of these multi-subunit complexes. So what we really wanted to know is, does TCP engage KDM1A in the brain? So to get to this, we used an assay that's been fairly recently popular, but based on very old concepts in drug development and in physics. And and that is a thermal shift assay. So for this experiment, we had two sets of mice. One were treated with rats. Two were treated with vehicle, two were treated with 15 mg per kg TCP. This is a high-ish dose, but if you adjust the mass to what a human dose would be, it's within the range that you would give someone with very treatment-resistant depression. The animals were exposed to compound. They were treated intraperitoneally, so systemic administration, for 2 hours. Then sacrifice and lysate was prepared from the grains. That lysate was heated to different temperatures and then centrifuged so that any proteins that had unfolded and aggregated could be precipitated in this step. And only thermally stable soluble proteins remained in the supernatant. And that soluble fraction is what we assay. And the basis of this experiment is this well-known observation that in general, proteins that are bound by a drug are more thermally stable than proteins that are the corresponding protein that is not bound. So here's what the data look like. You can see that animals treated with TCP have significantly more KDM1A in the soluble fraction resulting from heating to the indicated temperatures, and that this is actually a fairly large shift. So these data suggest that actually, although it's not that potent inhibitor in vitro, TCP does seem to engage KDM1A in the brain of systemically treated animals at clinically relevant doses. So that's cool. This suggests that perhaps some of the clinical efficacy of TCP is due to its inhibition of KDM1A. But that's really tricky to dissect because it's obviously also engaging the monoamine oxidases, as we've known about for decades. I just love this image. This is from a paper from the year I was born, from Jacob's postdoc mentor, Joanna Fowler, who established many aspects of PET imaging and is particularly well known for her work on monoamine oxidases. And so, unlike TCP, there are many compounds that do show selectivity between the monoamine oxidase A and B, and two of them are shown here. So in this PET image, you can see that both MAOA and MAOB are broadly expressed throughout the brain. So any molecule that has sort of low selectivity between KDM1A and these enzymes, there's no way we can really infer that much about KDM1A using that molecule. So what we really need is molecules with increased selectivity for KDM1A. So this slide summarizes a large amount of work by many people. But the big picture is that I was able to work with talented synthetic chemist from Mesh, former postdoc in Jacob's lab, and he prepared a series of compounds that are based on the core structure of TCP, but that are elaborated on either end. And this makes good biochemical sense because the active site of KDM1A is large compared to the active site of monoamine oxidases. It has to accommodate whole histone tails as opposed to just a small neurotransmitter. And I then worked with many scientists in the Haggerty lab to develop assays to profile the activity of KDM1A as well as monoamine oxidases and to compare the selectivity of these compounds in endpoint assays. And we ended up identifying one particular compound called RN1 that is about 1,000 times more potent for KDM1A than the monoamine oxidases. So it's kind of an interesting note on the side that in fact, the selectivity for the monoamine oxidases didn't really change that much. The reason it's a more selective compound is because we really drove down potency for KDM1A. So I took this compound and I asked whether or not we can measure changes in histone methylation. That's one of the main things we think about KDM1A doing. And despite a lot of different experimental attempts at this, I was never able to measure really consistent increases in bulk histone methylation at the genome-wide level. And this may be because in a cellular context, KDM1A is removing H3K4 methyl groups, but it's also being opposed by a lot of other enzymes like methyltransferases, so that the net effect might be compensated for. It could also be that the target genes of KDM1A are so few and so specific that you maybe wouldn't expect to see changes at the genome-wide level. But we wanted to check that the inhibitor, since we had modified the structure of TCP, was still relevant in targeting cellular KD1A. So to get around this, I developed a cell-free assay which uses immunoprecipitated KD1A from the cell nucleus, but still uses that synthetic peptide substrate. So now I've gotten rid of all of the opposing enzymes, and I'm simply asking, okay, full-length cellular KD1A in its complex, is it inhibited by RN1? And in this case, you can see by Western blotting that, in fact, you do have increased K4 methylation, suggesting that full-length KD1A is, in fact, inhibited by RN1. So we generated a very potent small molecule, but we wanted to further characterize its binding to cells. So this time not in animals, but I applied the exact same technique as before, the thermal shift assay in a cellular context. So now instead of rats here, I'm treating cells, then washing away the supernatant, and then going through a similar protocol where you collect the cells, you heat them to different temperatures, you pellet out the denatured and precipitated proteins, and you assay the soluble supernatant, and that the drug-bound proteins have increased thermal stability. And so instead of showing you a bunch of plots, I've condensed the results into a heat map, which I think is perfect, thermal shift. So again, sorry. You can see profound thermal stabilization of KD1A by both RN1 as well as TCP and another molecule that's related that I'm not going to tell you about today. So that checked out. Now, one of the things that I hypothesized we're gaining by having such a potent inhibitor is that I'm now able to measure changes in the thermal stabilization not only of KD1A, but also RPOR1. So RPOR1, I don't think is being directly targeted by KD1A, but that RN1 is conferring so much thermal stabilization to KD1A that the whole complex is in a sense being thermally stabilized. So I think an interesting future direction for applying this technique is to use it to analyze the complex specificity of small molecule chromatin binding targets, which is absolutely where the field is sort of going. Okay, so we have a potent KD1A inhibitor that engages KD1A. What does it do? So we worked with Marcelo Wood's lab. He's at UC Irvine, and his lab, they're experts in studying the behavioral effects of compounds generally, but in particular compounds that act on chromatin modifying proteins. And as just a first initial test, this is the first behavioral assay that was ever done using the KD1A inhibitor, we used a novel object recognition test, which I'll explain a little bit. This is busting up these whole psychology knowledge. So in this assay, you have a mouse and you put it in a chamber after you habituate it. And in the test chamber, there are two identical objects. So in this particular assay, the objects were a blue Lego or a beaker, and they were counterbalanced. So you let the mouse explore in the chamber, and then immediately after the training period, you inject either saline vehicle or test compound RN1. And then you exchange one of the items in the chamber for a new object. And then you return the mouse to the chamber for a 5-minute test. And you do this either 90 minutes after drug administration or the following day. And those are different routine intervals. So after 90 minutes, both the vehicle-treated animals and RN1-treated animals prefer the novel object. This is a well-established phenomenon, nice like novelty. So they will find the new object and spend more time investigating it. In contrast, a day later, the vehicle-treated animals still recognize the new object and care about it and want to go investigate it. Whereas the RN1-treated animals don't discriminate between the new object and the old object. So there's a few sort of ways you can think about this. One is that maybe the RN1-treated animals have impaired memory. Maybe they just don't remember which is the new object and which is the old. Maybe they just don't care. This is kind of a complex behavioral task. And anyone who's like worked with rodents will know they're pretty smart. It's hard to tell what exactly is happening. There's a lot of work that's been done to validate this assay using a combination of small molecule genetic techniques is definitely critical here, but it's not